All right, so I'm going to do an official welcome to our Chestnut Chat here in March. We're March 17th, and we're welcoming our poster session presenters from last year's symposium that was held in Asheville, North Carolina in the fall of 2022. As a reminder, for those of you who are new to Chestnut Chat or are just joining us and need a little bit of a refresher, our Chestnut Chat, we typically hold this year in 2023, we're going to typically hold them every other month. Although in April, we're going to hold a special dentata session. So mark your calendars for the third Friday in April, which, let me do some math, 1, 2, 3, that would be April 21st. We're going to do a special session regarding dentata for that Chestnut Chat. So many of you have heard about dentata. If you want to track trees that you've planted or seed or pollen that you receive, that is going to be the best way for you to do that. So if you've never heard of it, or if you want to know a little bit more about the program and the online software that we have to track tree planting, please plan on joining us in April for the dentata session. This is a webinar format for Zoom, and so as such, only the 6 people that you see here on your screen, me, Lisa, Trish, Kaitlyn, Morgan, and Kendra, we are the only people who can show our video and who can speak during the meeting. If you have any questions strictly for me as the technical person or want to chat with the fellow attendees, please use the chat function at the bottom of your screen. If you have a question for our presenters, I encourage you to use the Q&A function. That's how Kendra and I will keep track of questions to ask our presenters. So our format today, we're going to have Trish go first, and then we'll have some time for questions after that. Then we'll turn it over to Kaitlyn, have some questions after that, and then we'll turn it over to Morgan, who will wrap things up, and we'll have questions after that presentation. And we can also take any sort of at-large questions if we have time before 1 p.m. We always finish by 1 p.m. Even if discussion is lively and there are so many questions, we'll have to hold them for later. Okay. So chat is disabled. Happy St. Patrick's Day. Oh no. All right. Well, I will work on that because that's a new thing that I know I encountered in January. So I'm going to enable chat. While I do that, Lisa, will you welcome everyone here to the chat and then turn it over to Trish?
Sure will. Thank you so much, Sarah. It's great to be back on Chestnut Chat and see these amazing young conservationists present. They are our future, and we're so thrilled that you're willing to share your interesting research with everyone. So we need to build our conservation foundation with people just like you, and thanks to all the friends that I recognize on here. I want to call out Carl, Russell, and Anthony. You are really perfect attendance Chestnut Chatters, so that is just so great to see you all again. And I'm unfortunately have to not stay for the whole thing, but I look forward to watching the recording later. Going to take off for actually a long weekend. So Sarah looks like she's finished that. I don't know if Sarah introduced herself, but she is our Chief Conservation Officer, a very humble one at that. So I always try to give her a few props when I get a chance, not to embarrass her, but to let everybody know she's a really important leader in this organization, as is Kendra Collins, who's also on this chat. So we have a great team. It's going to be growing this year. We're hiring. It's a really exciting time in the organization with the documentary film launching on April 22nd. We're going to have a showing at the North Carolina Arboretum, and some of you are invited to that, local folks. We're going to be having screenings all over, up and down the Eastern Seaboard. So if you have ideas of a venue, we'd love to hear from you, because we're going to keep it kind of exclusive for a while. We're not going to just throw it out there to be screened or shared, downloaded, etc. quite yet, but it's really coming together. I'm very excited about it. We're also excited about the potential for the Darling 58 to be deregulated. So it's a very pivotal year for us, and I'm very excited to be a part of it. So with that, I'll turn it over to Trish, right, Sarah? Okay, Trish, take it away. And I'm going to say goodbye.
Thanks, Lisa. And then Trish, if you would just tell us a little bit about yourself, who you are, where you're from, and then launch on in. And after you're done, like I said, I'll take questions from the audience and feed them to you.
All right, sounds good. Does everyone see my screen?
Looks great.
Okay, sounds good. My name is Trish Nguyen, and I am a senior biology major at the University of Tennessee at Chattanooga. I've been volunteering with Dr. Craddock at his greenhouse since my freshman year, and I recently gave a flash talk at the TACF conference in Asheville. My project is titled A Small Stem Assay Using Hypovirulent Cryphonectria parasitica to Screen Castanea dentata backcross F2 families may set the stage for long-term survival. I hypothesized that the small stem assay using a hypovirulent strain of Cryphonectria parasitica will be able to show a statistically significant difference in blight resistance between the susceptible Castanea dentata and the resistant Castanea mollissima controls, because previous small stem assays have had enough resolution to distinguish between the two controls. I also hypothesized that the hypovirulent strain may be sensitive enough to rank each family of backcross hybrids by its level of blight resistance, because previous small stem assays that have used the virulent strain were not able to make that the distinction between each family. I planted the trees in mid-February and inoculated in mid-July. I inoculated the trees by snipping the top of the tree off at the point where the stem is 5 millimeters in diameter, and then I placed a bit of the hypovirulent fungus on the open wound. I measured the orange zone on each of the trees at 90 days post-inoculation in October. So in this first photo on the left, this is some of the greenhouse volunteers and me stepping up the trees from the small D40 pots that they were originally planted in into these bigger CP512s, and then adding a scoop of fertilizer to help them grow. In this middle photo, this is of a volunteer inoculating the tree. So this stem has already been snipped by Dr. Craddock, and then the hypovirulent fungus, which was grown on PDA, is stuck onto that open wound, and then it's all sealed off with a straw tip. And then this final photo is of me at 90 days post-inoculation measuring each canker. This is actually me measuring the full length of the canker, but in the end, I only chose to use the orange zone measurements, which is from the top of the tree to the farthest point down the stem where you can see orange growth. So with the help of Dr. Westbrook, I use the Duncan's multiple range test on my data with the outliers removed to see if any of the hybrid family averages were statistically significantly different from the controls. The SSA was able to differentiate between the two controls, the American and Chinese families, as expected. So the American family is in cyan over here, the CTEL 007 family, had the smallest cankers. Their cankers averaged to be about 9.6 millimeters. And for the Chinese, the AU-126, I'm sorry, I think I messed up. So American is cyan, it had 25.9 millimeters, and Chinese is AU-126. It had canker average canker length of about 9.6. I apologize. And then as for the intermediate hybrid families, they all range somewhere between the two controls, which was what I expected. But the SSA did not have enough resolution for me to distinguish between individual hybrid families. So it was all generally pretty blurry. I couldn't find one that was, I guess, like different from the rest. It had this level of resistance. I was not able to discern that. I used two different ways to visualize the data. So there was the canker rating on top and the scaled orange zone. The canker rating is on a scale of 0 to 4, with 0 being the most severe cankers and 4 being the least severe. And then the scaled orange zone is an inflated view of the range of averages with the Chinese blown up to 100 and the Americans scaled down to 0. That way you can see the averages of the hybrid families a lot more clearly. It's a lot more apparent. But even with these two ways of visualizing the data, I was not able to find a statistically significant difference in any of the intermediate families, the hybrid families. Dr. Westbrook also helped me create graphs correlating the known ancestry of the families with the progeny average blight resistance. So on the graph on the left, this is of the mother seed and dentata ancestry, so how much relation it has to an American chestnut tree. Here for both canker ratings and scaled orange zones, it's pretty strong negative correlation, meaning that it corroborates the idea that if you're more closely related to an American chestnut, you are more susceptible to blight. And on the right, this is of the C. mollissima ancestry, so the Chinese chestnut ancestry, and then the progeny average blight resistance. Once again, this is a fairly strongly positive correlation, which makes sense considering we know that if it is more closely related to a Chinese chestnut, it is less susceptible to blight. So what do I make of all of this? The SSA is not a good indicator for making selections within families. It can verify the degree of resistance from already selected hybrids, but I think that selection gains using blight, selection gains using the SSA in blight resistance will be very low because my results showed that the estimated heritability of blight resistance is low. And then I think another possibility as to why my SSA had such low resolution could be because I used the hypovirulent strain. So the strain of Cryphonectria that I used is called the weekly strain, which already has fairly low pathogenicity. And then coupled with the fact that it's also hypovirulent makes it even weaker. So with such weak fungus, I think that the growth in the cankers was not drastic at all. It was hard to distinguish between each of the individuals because of such a weak fungus. I think for better resolution in future SSAs, maybe use more trees but fewer families. So in my experiment, I had about 300, which was a decent workload, but I think maybe in future SSAs use fewer families and then more trees. I had 13 families and then about 28 trees, so maybe 3 and then scale up the amount of individuals in each family. And then also maybe stick to a virulent fungus. Maybe the hypovirulent wasn't strong enough to show any difference, so the virulent fungus might be the way to go. Thank you.
Thanks so much, Trish. I have a question about your observation of the cankers and if you think that scaled orange zone was, and I know you had a comparison between a strict rating of that 0 to 4 and then you had the scaled orange zone. Was there anything that you noticed on the cankers like callus development or sporulation that seemed remarkable? And I recognizing that you didn't take those data intentionally, but I wonder if just from your observation or your recollection, would either of those have been potentially additional or better measures of resistance?
I did look at canker swelling. I mostly just noted whether or not there was swelling. It was a little inconsistent from what I saw. I think some trees had swelling, like all of the orange zone was, I guess, swollen, and then others didn't really have much swelling at all. I'm not sure. I looked at it, but I did not really like quantitatively measure it. I just noted the morphology. That's about it. I wish I had a better answer for you.
Oh, no, no problem. And then one more question regarding hypovirulence, because I have not actually done any inoculations with a hypovirulent strain myself. In the ones that we've done here at Penn State, it's all been either SG23 or EP155, and EP155 is a very highly virulent strain. And that, I mean, that's bringing the hammer down, and most of them croak really quick. And then the SG23 is a fairly weak strain, not as weak. I actually think it's a little weaker than weekly, but I'm not sure because I've never compared them directly on my own. But I wonder, in terms of like that callus development, did you, and I think you even mentioned this, but I wonder if you could expand on how badly the Americans performed. You know, did you see any swelling at all from those American controls because it was hypovirulent, or were they fully a sunken and fully orange zone canker on those Americans? And then what did you see? What, because it was hypovirulent, did you see all the trees being able to evoke some sort of resistance response, or were the Americans toast regardless because it was hypovirulent, and then you had more callus development on up? And you may not be able to answer that question, but I'm just curious.
I think with the fungus that I used, I don't think any of them are toast. I had one tree I remember specifically that was runty, and then it was like completely overtaken by blight, but that was just 1 out of like 300. I think the hypervirulent fungus gave it a better chance at surviving just because it's weaker. Yeah, I'm not sure about swelling or like sporulation. I didn't really look at that. I also only used the hypovirulent fungus on this experiment. I didn't compare virulent growth versus hypovirulent growth. So if I want to answer that, I'll probably need to look back at previous literature. I know one of Dr. Craddock's previous SSA students used the virulent fungus on her SSA, but I have not compared the two together, and I don't have that data here. I'm sorry.
No, I didn't expect you to. So just curious your reaction. So no problem. We have a question from Jeffrey, who asks, are you continuing to follow the seedlings, or have they all been discarded?
So we cut off all the cankers just to make sure it didn't spread. I'm pretty sure all the trees are still alive. I haven't been back to the greenhouse since spring break started, but I think the trees are fine. If that's what you're asking, we cut off all the cankers just to make sure that the blight didn't spread unnecessarily.
Is the plan to plant those out in the field? And because I think in a lot of these modified SSAs, the goal is to do an SSA in the greenhouse, plant them out, and then do an in the field inoculation to see if they match what we saw in the greenhouse. Do you think that that's maybe what the next step is?
Yes. Dr. Craddock mentioned that the trees will be planted in one of the orchards in Middle Tennessee. I'm not sure if that's been done yet, but that's definitely going to happen in the future.
I saw Kaitlyn nodding her head, so I imagine that might be a future step. So awesome. Well, Trish, great work. Thank you so much for sharing that and putting this talk together and being willing to participate in our first poster session presentation, Chestnut Chat. I hope this is the first of many to come in the future. All right, thanks so much, Trish. Kaitlyn, you're up. If you can tell us a little bit about yourself. I see some clapping hands, yay. So Kaitlyn, if you can tell us a little bit about yourself, who you are, what makes you tick, and what your talk is going to be about.
Can you hear me okay?
Yes, I can hear you. And
Okay.
if you want to share your screen.
Yes.
More clapping hands, yay.
So, present. Let me switch these.
You're on it, Kaitlyn. Good job.
Yes.
Yes,
Okay,
perfect.
so
thank you.
My name is Kaitlyn Harden. I'm a senior biology student at the University of Tennessee, at Chattanooga, like Trish, and I'm also a student of Dr. Craddock's. I've helped at the greenhouse, volunteered for about 2 years now. And the poster I presented in October showed my early findings within oxalic acid leaf disc assays. The title was Oxalic Acid Leaf Disc Assays may be another method of screening phenotypic variability in American-looking Castanea hybrid trees. They're important because they might be another screening method in the future. And this is my undergraduate thesis, like Trish. I'm completing it this spring, so I'm going to share some more detailed results than my preliminary results than that I showed this fall, or this past fall. Here's me with some of my tubes. Dr. Craddock took this picture, probably wanted me to include it. As you can see, I very haphazardly taped all the tubes on the table most of the time. It was very fun. Anyway, some background information. Bennett and Hindle in 1989 noticed the relationship between oxalic acid and Cryphonectria parasitica. Oxalic acid is used by many pathogenic fungi to weaken the cell wall. Oxalic acid acidifies host tissues and sequestrates calcium from the host cell walls. Cryphonectria parasitica effectively uses oxalic acid to kill Castanea species. By studying oxalic acid's effects on several lineages, we may be able to characterize trees with a relative tolerance to oxalic acid. The acid causes browning of leaf tissue. And biotechnology, the newest way to fight the fungus is with the oxalate oxidase gene from wheat called OXO. Right now, our screening method for container-grown seedlings is a small stem assay that Trish used and Morgan also used. They offer the promise of a quick, effective, and inexpensive screening process for selecting blight-resistant trees. They're infected at a young age with the fungus at the greenhouse, and then they're measured on their visibly infected areas. But there's a few issues with the SSA. The fungus can be applied inconsistently, which causes no takes, and then they're not measured or considered in the study. And then later on, when they're planted in future orchards, they might not thrive because they've been previously inoculated with the fungus. So the role of oxalic acid in this pathosystem led to the first attempt to use OXO in genetically modified chestnut trees. As we studied the transformed Castanea with the OXO, we may be able to characterize trees using relative tolerance. We can use this knowledge to characterize our hybrid germplasm and seedling populations that we expect to vary. This is just an image of some of the GMO chestnuts and how they react to blight. My hypothesis, I hypothesized that the two control C. dentata, the American blight susceptible tree, and C. millesima, the Chinese blight resistant tree, will show statistically differences in their oxalic acid tolerance. The hybrid families used in the small stem assay showed intermediate tolerance. This was shown in measurable differences in leaf disc necrosis following an oxalic acid soak of the leaves of several species of chinkapins, C. dentata, C. millesima, and open pollinated seedlings of selected backcross hybrids. The resistance of C. pumila species to C. parasitica are understudied, and I thought their endogenous tolerance was going to be somewhere between C. dentata and C. millesima. So I used 308 trees in my study. Leaves from all 308 sample trees were collected within a 24-hour period. The fifth leaf from the tip of the stem was collected, just to keep everything, the same. One leaf was collected for each tree. So there was only one leaf per tree. I know these replications and observations can get confusing, so I like to reiterate that. And I used the same trees from Trish's small stem assay. So it's the same families and the same trees from her small stem assays, except for the chinkapins. Those are the ones that were available to me at the greenhouse. One leaf from each individual tree was used to create 10 discs. The discs were 1.5 centimeters and cut from a metal hole punch. The discs were placed in 50 millimolar oxalic acid solution in a closed tube. And then, like in the picture, they were taped horizontally onto the orbital shaker to prevent the disc from clumping. I took images of all the discs after the 10-hour soak, and then I measured them using image J. But with the oxalic acid leaf disc assay being a new idea, many preliminary trials were completed to determine the right configuration for testing. First, the age of the leaf was determined to affect the soak. One leaf was collected that was fresh, which I considered was like the first fully expanded leaf from the tree. One from the middle of the same tree, and then one from the very bottom of the tree. Both the old and the new leaves reacted better than the middle leaf. So reacting better, in my opinion, was a more distinct brown-green area on the actual disc. So I used the fresh, fully expanded leaf for all my experiments. Another test was for the time duration. When I first began this, Dr. Nuha suggested that I run these for 12 hours. But when I started doing that, the American chestnuts were completely brown within the 12 hours. So I ran a trial where I checked them every hour for 12 hours, and 10 hours seemed to be the threshold before the hybrids and the Americans turned completely brown. So I conducted these soaks for 10 hours. And then I kind of tested to see if the inversions of the tubes on the orbital shaker affected anything, and they didn't. So I did horizontal because that was the easiest to get them all on there. Anyway, here is a picture of the discs before they're soaked. They're small. Yeah. Here they are taped on here. Once again, I just kind of put them where they fit. And then here's an example image after an assay. It's not glamorous, but it worked for image J. The ruler was needed for scale in image J, so you can measure the discs. This tube is just for the tree ID in our small stem assay, and all I needed to see was the distinct brown versus green tissue, and I can measure each individual disc that way. And this table shows the 18 families used in my leaf disc soak assay, and 13 of the families come from the small stem assay that Trish used. So those are my large sample sizes. My chinkapins have very small sample sizes because I can only use what was available to me. Yeah. Well, I should probably keep on this so you can look for a little bit because I put the seedling types. That's interesting to note. And then here are example photographs for each disc that are representative. So I found one that was like the median for each family. So this one is Chinese, C. millesima, and this one is American, C. dentata. So slightly different. I think it's interesting that even the browning is different. And then the next 11 are the hybrid families, and you can't really tell them apart at all, even visually, which is not a good sign. And then the chinkapins are the last 5, which are astonishing. That was the weirdest result that we have gotten from this study, because previously thought, chinkapins are susceptible to blight, but why are they tolerant to oxalic acid then? But I guess another thing to consider is that I had to include the mid vein in all the chinkapin leaf discs. So that's something that was not included in the rest of the leaf discs because the leaves were a lot bigger, but these were so small I had to include the mid vein. So I feel like that affected it somehow. Anyway, here is a graph generated in R. It shows a box plot for each SSA family. The first is C. millesima again, the Chinese. The second is C. dentata. The difference is measurable. Hybrids fall in between. But with the hybrids, there's major outliers in a large range. I suspect this has to do with their genetic variants and further proves my later point of oxalic acid tolerance being multigenic. And then here is a smaller graph of just C. millesima and C. dentata compared to the chinkapins, which all have a lower median compared to the other Castanea species, which once again, I just want to reiterate that that is a very abnormal finding and it needs to be studied further. I can't do a lot statistically with it because my sample sizes were so small. Like, I mean, Ozarkensis, I had one tree. Yeah, so they're wildly understudied, but I think there's something going on and we need to figure out what's happening. And in R, ANOVA showed that my results are significant for the SSA. Once again, I didn't include the chinkapins because they're so small and it would feel like it would ruin it. And then Dr. Westbrook also helped me with this one. So the top and the bottom graphs are Trish's, and mine is this middle one. So we see a high positive correlation between progeny means, mother C. dentata ancestry, and oxalic acid necrosis. So that's interesting. And the increase in oxalic acid necrosis and C. dentata mom ancestry suggests that oxalic acid tolerance is a multigenic trait in the Chinese chestnut. So yeah, as expected, the tolerances of the hybrid family stay between the controls of C. millesima and C. dentata. High positive correlation between progeny means, mother ancestry, and oxalic acid necrosis levels. As C. dentata ancestry increases, oxalic acid necrosis increases. Oxalic acid tolerance might be a multigenic trait. Also, there's more at play in the pathosystem. C. pumila is so tolerant of oxalic acid, but still susceptible to blight. So we may need more biotechnology in the future to fully restore the American chestnut. But C. pumila will be studied further in this year's SSA and oxalic acid studies at the Tennessee chapter. We have them included in the SSA, so we're going to see how that goes. And another thing about this screening method is that oxalic acid is very accessible. Like you can buy this at Ace Hardware. I did before my lab grade chemicals came in, just to like mess around with it. And so it had me thinking, I'm like, is this a screening method that we can use citizen science for? You know, it's just something to consider. Because it's really easy. I mean, like the actual science experiment is like elementary level, it feels like. And then I presented in Dallas, and it was kind of sad because no one knew what chestnut blight was. Like literally not a single person who came up to me knew what chestnut blight was. One man from New York just said, "I don't think we have those in New York." And I was like, "Oh, you absolutely do." So yeah, interesting. But I defend my thesis on March 31st. So, and then here's my references. but I want to give a huge thank you to Dr. Jared Westbrook for his immeasurable help with the statistical analysis in R, and Dr. Newhouse for his help with the early stages when I began to develop my methodology. I also want to thank the UTC Honors College for funding my research, and the American Chestnut Foundation for giving me the poster award in October, and for allowing me to share my results to more people. And here's my contact information. Yeah, I guess I'll answer questions though.
Thanks so much, Kaitlyn. We're getting clapping hands, and I saw an old face for how can people in New York not know that there's like millions, like literally millions of chestnuts across the state. So you are an ambassador, especially taking it down to Texas, where there aren't millions of. In fact, I don't know if there are any dentata down there. There may be some of Arkansas and Texas. Hill would know better, I think, on that one. We have a couple questions here. I'm because I'm the host, I get to ask my questions first. So I'm going to take that liberty. Kaitlyn, did you do any multiple discs of the same tree to make any comparisons? Because I know you said maybe you only had one, it was Arkansas, but did you do any comparisons of many discs from a single individual to see what the range of response might be there?
No, because I did 10 discs per tree, and I think I forgot to state that in because I was too busy reiterating that it was one leaf per tree. So it's one leaf per tree that makes 10 discs. So there's 10 measurements per tree.
And then what was the range of response in a given tree? Because what you were showing us was the average across individuals per family, but what was the range within a given tree? What was it? Was it extreme as well?
Yeah, the range was extreme, which I'm surprised it works statistically because the range was so extreme.
Yeah. And so Marty asks, and I think this is a good follow-up to that, could you use stem samples? And what do you surmise would be the response with stem tissue versus leaf tissue?
Yeah, I don't know, because I think we use leaf tissue because it's easier to see the color contrast. I'm sure we could give it a shot to see what stem tissue will do. That's something I can pass on to Uma, the student who's taking over this project, and see what happens. Yeah.
That might be interesting. And another personal question: what is oxalic acid? Why do you find that at Ace Hardware? And what are other people using it for? Because probably not chestnut leaf disc assays.
No, it's advertised as like a wood bleach or sometimes a cleaner. I found it in Barkeeper's Friend too. Like the kitchen cleaner. Yeah, so it's in a lot of cleaners actually.
Nice, good to know. All right, Carl Absher asks, in my area, it seemed that chinkapin survived, so that would be Southwest Virginia. It seemed that chinkapin survived longer than the chestnuts, well into the 70s, and there are more of them still around and still flowering. Could the oxalic acid tolerance contribute to that, if it is truly so? And could it indicate a lot more to blight resistance than oxalic acid detoxification?
It might. I mean, the thing about chinkapins is that they're like wildly understudied. So we'll just see what happens in the small stem assay this year and what we can conclude from that. But yeah.
How many chinkapins are in the SSA for this year?
I don't know exactly. That would be a Dr. Craddock question.
Okay. All right. Is he on here? I haven't seen him. Hill, if you're here, maybe. Let's see. There he is. Maybe Hill can throw that in the chat. All right. Ron asks, is anything about genetics of oxalic acid tolerance in chestnut or centennic species? Is anything known about the genetics of oxalic acid tolerance in chestnut or centennic species? Where might this trait map relative to chromosome positions of known chestnut or other centennic R genes? I don't know that we know.
Yeah, I don't know.
Let me see if Jared's on here. He may not be. I don't see him on here. He would be the person who would know, and I have not heard of anything that we know about that, Ron. I mean, as far as we know, there is no specific oxalic acid. Well, I'm not even going to surmise. I don't know.
Yeah.
Steve Jeffers says, be careful with interpreting results because there was only one leaf tree used. In the future, I suggest using multiple leaves per tree. I think we established that you did, right? That you did 10. Oh, well, no, you only used one leaf.
One leaf.
It was 10 samples
Right.
per leaf. So Steve's
Yeah.
saying maybe use multiple leaves per tree.
Yeah. And that's something we can consider in the future. This is a first time running this.
Yeah. Okay.
screening,
Thanks, Steve.
So yeah, thank you.
All right. Well, great job, Kaitlyn. Thank you so much for being with us today and sharing your work. Morgan, you are up. Oh wait, hold on. There might be one more question. What's it say? Marty Jessel says oxalic acid is used to control varroa mites in honeybee hives. I did not know that. Maybe that's another. Yeah. All right. Well, thanks for sharing that, Marty.
Yeah, the tube or the package I had, it's just like the dry powder and it just says wood bleach on it. So yeah, that's what it was advertised for me. So
okay. Well, it seems to have a lot of uses. We'll have to, what else can we use oxalic acid for? All right, great. O'Hill says 100 seedlings per species for Pumila ozarkensis and wow, that's a pretty big undertaking. Thanks for writing that out, Hill. Okay, Morgan, if you can take it over, introduce yourself, who you are, what you love to do, chestnut or otherwise, and take it away.
Awesome, I'm glad to be here. So I'm Morgan Wingo. I go to Berry College and I work under Dr. Cipollini, and we've been working on some small stem assays as well. Okay, so the poster I presented when I went to Symposium was the validation of an alternative small stem assay for blight resistance in backcross hybrid chestnut seedlings of the Castanea genus. Given that blight resistance is a polygenic, or it's well understood to be a polygenic inheritance, it's unlikely that when trying to get backcross seedlings, we're going to get much higher than an F1 level of resistance at this point in our progress. And so it's increasingly important to have an assay that is able to discriminate between these lower levels of resistance so that we can move forward with the program. Progeny tests are done for two different reasons, for backward or forward selection. With backward selection, we're selecting the parents for breeding based on the average resistance level of their offspring. So the superior lines of offspring that have higher levels of resistance are used to identify superior parents. Those parents of these lines are potentially possess a greater number of on average of the alleles that it confer resistance, and it's likely that they're conferring that to their offspring. So breeding them will allow us to produce more offspring in the future that could have high resistance. With forward selection, we're selecting progeny to keep or to cull based on the level of resistance relative to others within their line. So with backward selection, we're looking at a line as a whole, and with forward selection, we're looking at individuals within the line. We'll pick the most resistant of each line, and they're selected for continued use in the program. So we'll outplant them, allow them to grow and conduct subsequent orchard inoculations, and then potentially include them in the breeding program. And this is the primary intention for the ALT assay. So there are a few assay methods. I mentioned orchard inoculations. Before starting to use small stem assays, we had to wait 4 to 5 years before we could conduct any kind of inoculation on the tree, because we had to wait for the diameter of breast height to get to about 2 inches in order to not just obliterate the tree. With this method, we take a cork borer and we remove a bark plug, and then we replace it with an auger plug that has the Cryphonectria parasitica inoculum on it. We place the mycelia side down into the hole and then wrap this in tape in order to seal in moisture. This is still often aggressive and can be lethal to small stems. And we have to wait a long time to be able to do it. So in order to speed up the process, there's the standard small stem assays that are used on first year seedlings with approximately 3 millimeter diameter stems. With this, you make a vertical incision in the stem and you insert the auger plug with the mycelia and then wrap it with the parafilm to seal in the moisture. This technique or this method can be technically difficult. It's hard to induce consistent cankering as a result of the small area that you're trying to push mycelia into. So it's not always, they won't always take and don't always produce a canker that's representative of the resistance of the seedling. With the alternative small stem assay, we were kind of aiming to try to increase the consistency, among other things. We conducted this on first and second year seedlings of approximately 4 to 5 millimeter diameter stems. And in this case, you snip off the top and place an auger plug with mycelia side face down to flush with the exposed tissues, and then seal it with a half sealed straw. After about a week, we'll remove these straws and allow the cankers to continue to grow for about 90 days, after which we'll measure the cankers and then remove those cankers and disinfect the remaining stem with a misting of 70% ethanol. Canker removal and seedling survival is pretty successful with the alt-S assays. However, the timing of the inoculations is pretty important, that we do it around midsummer, and this is in order to avert apical dominance early enough. This encourages the new shoot development lower on the stem so that vertical growth can continue to happen even after cankers are removed and seedlings are better able to recover. The tests are able to produce healthy seedlings that haven't been majorly damaged outside of where the canker occurred at the very top of the stem, and these seedlings can be successfully outplanted into orchards. So to validate the efficacy of to find the validity of this test, to see how well the assay does at discriminating resistance within seedlings, we measured a variety of phenotypic variables that we know are representative of resistance. So orange zone canker length and full zone canker length, as mentioned, and then as well as shrinkage, swelling at the base, and then in the 2021 assays, we did sporulation as well. We ran these through a principal component analysis in order to try to reduce the amount of variables that we had to work with. And the PCA identified that the first axis was able to account for 56.5 and 36.5% of the variation that we see in the seedlings. So this first axis is in a way replacing all of these variables to create a more succinct evaluation of resistance. And then on that axis, orange zone canker length had the most loading, meaning it was contributing a lot of understanding about the variation that was going on. So with that information, we considered that there was potential for the orange zone canker length to serve as a proxy for a level of blight resistance when running these assays. So we derived resistance scores for the seedlings based off the first axis of the principal component analysis, which is on the y-axis, which shows you a good vertical distribution of Chinese at the bottom and American at the top, with our hybrids distributed in the middle. And then orange zone canker length, the mean orange zone canker length of the families that were included in this assay on the x-axis. And so having this horizontal spread along with the vertical spread that was derived from the PC1, it shows that the orange zone canker length is able to relatively show what we want to see, this distinguishment between Chinese and American, and some kind of spread between different backcross families. To further determine if this orange zone canker length could really do this, we then compared it to predictors based off of parental phenotypes. So we took phenotypes of the parent trees involved in creating these lines, and these factor descriptions represent positive signs of blight resistance. So if it's not succumbing to cankers, if it's not succumbing to the blight and it's making good cankers, main stem will still be alive, high percentage of the canopy will be alive, callousing is present, suggesting it's preventing the fungus from spreading. And so these phenotypes were then used in this PCA to see that the first axis accounted for about 38.5% of the variation in resistance of the parent trees. And we derived, again, a resistance score or resistance scores for the parent trees using this first axis of their PCA, and regressed it against the orange zone canker length for the seedlings. Again, we see that we have a nice spread from Chinese to American, with the back crosses in the middle. With the orange zone canker length on the vertical axis, we can see some more distinguishment, not only to the left and right, but up and down. So again, the parental predictors were aligning with what we think the OZCL is showing. Then with the help of Jared Westbrook and others, we were able to develop blight resistance indices based off of genetics of the parent trees. So we created a genetic blight index for the parent trees and regressed it against the orange zone canker length. And though it's flipped with American on the left now, we're still seeing that same kind of distribution that we want to see. These Chinese on the far right suggesting high resistance, with Americans on the far left suggesting low resistance, and the spread of backcross in the middle. And last, we took again with the help of Jared Westbrook and others, we created an index based off the percentage of American ancestry of the parents. And what's interesting here is that with backcross, the idea is that we're getting a higher percentage of American while still trying to confer more resistance, like Chinese-like resistance. And so on the x-axis, you see the proportion of American ancestry is high, so all of the backcrosses are clumped over to the right. But with the y-axis, the mean orange zone canker length allows some more discrimination to be seen, with some of these backcrosses being closer to Chinese resistance and some of these backcrosses being closer to American resistance. So that's a distinguishment between the lines that we're able to get with those orange zone canker lengths. Another important aspect of these assays were that we were able to get high survivorship after outplanting. On the left of this image, or this image is from a follow-up we did to one of our orchards in Ellijay in May of this year. On the left are plants from the 2020 assays outplanted in 2021, and on the right are 2021 assays outplanted in 2022. We had 77% survivorship at this point for the 2021 outplanting and a 70% survivorship for the 22 outplanting. For the 2021 outplanting on the left, the losses were evaluated to be a result of natural basal cankering that was unrelated to the initial inoculations themselves. And on the right, it was suspected that the losses we had there were a result of a late freeze after leaf out. So what losses we saw were assumed to not be directly related to the assays themselves, but other consequences of the field. Important results of running these assays has helped us with the validation of the method. We're able to detect a full blight resistance range that should be expected among family lines. So we're seeing Chinese and American at the far terminal opposing ends with the backcrosses spread between. We're seeing the resistance values ranging from F1 intermediate level to American low level resistance, which is expected of backcross lines. And the results of these assays, as evaluated by the OZCL and other phenotypic variables, agree with the predictions based on the genetics, ancestry, pedigree, and phenotypes of the parent trees that were involved in creating these seedlings. The orange zone canker length does seem to reliably represent blight resistance, and removal of the cankers results in relatively undamaged and healthy seedlings, which is an important step forward from the standard assay, which could often make the seedlings not able to be successfully outplanted. This image on the right is a good comparison of the cankering you see between American-like and chestnut-like seedlings, with the left side being American. And the O arrow is the orange zone canker length, and the F arrow is the full zone canker length. It's suspected that the orange zone canker length might have to do with the depth at which the fungus is able to penetrate into the tissues. That's an ongoing understanding that we're trying to work with or work through, but it's clear that Americans have much larger cankers and Chinese have much smaller cankers, and this orange zone canker length seems to be more consistently proportional to the resistance of the tree, as opposed to the full zone canker length, in which outside of the orange zone, what is left as full zone canker length is about the same between these two trees, whereas there's a large amount of variance we see with the orange zone between resistance. Moving forward, in the short term, we intend to continue to utilize these alt SSA in our nursery as a screening method and continue outplanting seedlings selected with this alt SSA. We conducted more this past summer 2022 and outplanted them just a couple weeks ago. In the long term, we plan to return to these these outplantings and evaluate these trees that were outplanted as alt assay seedlings once they've gotten to large enough size, so approximately 2 inches in diameter at breast height. And then we plan to compare the results of the alt assay to the results we see from these inoculations. Recommendations for its expanded use is that across more programs, the discriminative ability as a screening method is utilized. And this allows for the removal of the most blight susceptible individuals with the potential to accelerate gains in the TACF breeding program via early forward selection. This discrimination and elimination of non-resistant or less resistant individuals allows for lower initial planting densities into the orchards, which allows for wider spacing and better plant care with the potential of increasing survival and vigor in the field. It also saves time and resources that are spent on growing trees that are never going to make it. This potential still remains under evaluation, but those are definitely the hopes. And that's it. Thank you.
Excellent. Thanks, Morgan. Clapping hands. Yay. I love like these emojis and everything that make me very happy. I have a question regarding the outplanting, because here in Pennsylvania, that is tremendous growth from those 2021 plants. What sort of magic are you doing on those trees to make them grow so fast? And can we have some of it? Or is it just because Georgia is so warm?
I wish I could tell you. I'm not entirely sure. There is the steward of that particular site that we continue to outplant into, and I'm sure he's doing great work. It might be that Georgia being more south is just a little bit more conducive to faster growth rates, but I'm not entirely sure.
Well, I hear in 80 years here in PA, we'll have Georgia-like weather. So maybe I just got to wait 80 years. So, I'll think about that. All right, let's see if we have any other. I don't see any other questions. Morgan, what do you think about your results versus what Trish saw in her evaluations? Do you have any thoughts about comparing what you saw? It seems like everything's pretty similar. That I don't see anything that was striking that was necessarily remarkable between. But I wonder if you had any thoughts of what Trish saw versus what you saw.
Well, I think, as she suggested, that using the hypovirulent strain might have just been the reason that less discrimination was possible, because we used the EP155 and the SG123 as for our inoculations, which are more virulent than the weekly strain, even when the weekly isn't hypovirulent. So I think that is most likely why we were able to get better discrimination ability, because regardless, outside of what strain you're using, the assay method of cutting the tip as opposed to making the vertical incision does allow for more consistent cankering. So I think it's just going to depend upon the fungus's ability then to really take hold and do what it needs to do. Okay,
makes sense. Well, I don't see any other questions. I'm going to delay a little bit to see if anybody wants to ask any questions. I get a lot of questions about this. People ask, are these balloons? Are you having a celebration? And I'm not. These are big beach balls for my kids who are 5 and 7, and I have to keep them out of reach, or else they throw them at the TV or me a lot. So those are beach balls. I've had people ask, is this a bathroom? It's not. That's the doorway to my kitchen. So maybe it's because they're like the weird like doors. I don't know, but that's my kitchen, and that is not toilet paper. Those are paper towels. So hopefully that solves some mysteries for our regular Chestnut Chat attendees. Oh, we've got question. It worked. Jeffrey asks, can you comment on the AM1 versus AM2 parent? They seemed rather different, including AM2 being better than some of the families.
Here, let me share my screen again. I'll go back to the PowerPoint. Is he referring to the phenotypic analysis of the parents?
I don't know. I think just talk about anything that you want to, and maybe he'll elaborate.
Sorry, the little Q&A boxes was in my way. I think here he's kind of referring to the clumping between these 2 years. It's just these were, with different years, different lines were bred, and not all of the same lines were in the 2021 assays were in the 2020 assays. We used, I believe 35 families overall, but not all families that were in one were in the other. And they were different seeds. They were different productions. We harvest our seeds from regional orchards, leave them in the, process them and leave them in the fridge until spring when we plant them and grow those seedlings. And then we conduct our inoculations later that year. And so it's likely that just a different kind of a resistance was inferred in those seedlings or conferred into those seedlings. And I'm not entirely sure if we know whether it has to do too much with the parents or if it's
Marty says that there were small sample sizes for the Americans too. So that seems to be a big issue. Where are the Americans from? So they're from Georgia though? Can you say that for sure?
I believe that all of the seedlings we use are from Georgia.
Okay.
Georgia has a high amount of diversity comparative to some of the other areas in the range. So that was another aspect of trying to see if this assay could really see the diversity that we have genetically in our range.
Okay. And then Paul asks, or he states and asks, you mentioned that calcium levels helped. Could this play a difference? And he mentions that he has a very high calcium level in his soil. Can you mention any more about calcium and the role that it might play?
I'm not overly familiar with the roles of calcium cycling in chestnuts. I know that it can be a very important nutrient, but I don't believe we've done any particular soil sampling in the orchards that we've been out planting into. So I can't make too much comment on that. I'm sorry.
Yeah, nor can I. I'm afraid I don't know much about that. Although we're hoping to get some more. We've been running some tests with Americans, Chinese, backcross, and particularly the transgenic trees, looking at nutrient cycling, and hopefully we have some better answers in the next couple of years after we do some leaf and other types of analyses. Marty says most orchard sites are low in calcium. Seed dentata is sensitive to high calcium. Steve Jeffers says, does the straw cap limit the size of the stem to be tested such that only stems with the exact diameter can be used?
And actually, we kind of ran into that problem this most recent summer. We ended up having to order larger straws because some of the second years we tested, there wasn't an area where we could get a small enough route, about like 4 or 5 millimeters. So I think we ordered 6 to 7 millimeter straws. And so then we would just, we had pre-prepared a good amount of those and would just assess which one we needed to use when we put them on.
Yeah, we've done the modified SSA here at Penn State a couple times, and yeah, you have to. It depends on where you can cut the stem. We were able to cut off. The Chinese were always the biggest, and we attribute that typically because they're larger nuts and you get larger seedlings typically in that first year of growth, and we always had to cut them up real high so we could fit the straw on top.
I will say that alternative to using parafilm with the alt SSA, we had. At sometimes if the straw still wouldn't fit, we would have to use parafilm and wrap it around it. And that is still very technically difficult to keep the auger on top of it and not lose it while you're trying to wrap the parafilm around it. So it was nice being able to just order a slightly larger straw and just slip those on top of it.
Absolutely. Lindsay says, any thoughts about if using these small stem assays would be possible on twigs from more established trees already in orchards? Or is this mostly useful just for young seedlings?
I don't see necessarily why it wouldn't be a helpful tool. In fact, I mean, I know that orchard inoculations, since they're done on the trunk, they can often be pretty damaging to the tree. Even when it seems like the tree is showing relatively good resistance, sometimes it just doesn't pan out well. And so potentially conducting small stem assays as orchard inoculations as an alternative to trunk inoculations could have potential, but I'm not entirely sure.
So Marty says we have tried it, lots of variation among branches on trees, and the results have less discrimination, which I think we've seen that too. So I have not done and have not seen doing the modified SSA where you snip it off and put the thing on top and put the straw on, but we've done old school, scratch it, put the auger in, and there's something about branches that are screwy, and I'm sure there's some physiological, and some physiologists could probably school me on why that is, but yeah, we tend to get much wider confidence intervals and range of results on branches than we do on the main stem, which is why we use that. But you're absolutely correct that it can be a very damaging assay to do it. You just stick in, especially EP155, you take a really highly virulent strain and you throw it right in the main stem. That's not good for the tree. But you can cut it off at the base and let it resprout, but still it's typically not as hardy as the original stem that came up. So great question. Thank you, Morgan, Trish, Kaitlyn. I really appreciate your time. And to all of you who were able to join us today, I really appreciate that. So I'll say again, in April, we're going to have a special dentata edition. We don't yet have a topic for May. We have a couple different topics that we're playing around with, but I can't say anything yet because we haven't identified it yet. I will say our November date is established. We're going to be joined by Michael French, who will be talking about mineland reclamation and restoration with chestnut and other species. So keep that November date in your calendars, and that topic is definitive. So until April and or May, I hope you all have a marvelous spring. And Morgan, Trish, Kaitlyn, any other final words?
No, just thank you for having me.
All right. Well, thank you so much for taking the time, and yeah, see you guys in a future edition.